Welcome students. Last episode we discussed the stability of DNA. In this episode, the organization of genome in prokaryotic and eukaryotic in detail. First of all, what is meant by genome and what is meant by genomics? The word genome, coined by German botanist Hans Winker in 1920, was derived simply by combining gene and the final syllabi of chromosome. It is not specified. Genome usually refers to the nuclear genome. An organism genome is defined as the complete haplogenetic complement of a typical cell. The genetic content of the organelles in the cell is not considered part of a nuclear genome. In diploid organisms, sequences variation exists between the two copies of each chromosome present in a cell. The genome is the ultimate source of information about an organism. Genes are units of genetic information present on the DNA in the chromosome and chromatin. Genome is all the DNA contained in an organism or a cell, which includes the chromosome plus the DNA in mitochondria. The number of genomes sequenced in the entirely is now in the thousands and includes organisms ranging from bacteria to mammals. The first complete genome to be sequenced was that the bacterium Haemophilus influenza in 1995. The first eukaryotic genome sequence, that of the yeast Saccharomyces cerevisiae, followed in 1996. The genome sequence for the bacterium E. coli became available in 1997. The much larger effort directed at the human genome was also accelerating. The differences between the prokaryotic and eukaryotic genome are: in prokaryotic, single cell genome is present, and in eukaryotic, single or multi cells are present. In prokaryotic, no nucleus is present, and then in eukaryotic, nucleus is present. In prokaryotic, one piece of circular DNA is found, and in eukaryotic, chromosomes are found. In prokaryotic, no mRNA post-transcriptional modification is occurred. In eukaryotic, exons and intron splicings are occurred. Genome organization. A genome is an organism's complete set of DNA comprising of nuclear and mitochondrial DNA. Each genome contains all of the information needed to build and maintain that organism. A human haploid cell consists of 23 nuclear chromosomes and 1 mitochondrial chromosome, contains more than 3.2 billion DNA base pairs. Prokaryotic genome organization. Prokaryotes were the earliest organism on Earth. Today, they still dominate the biosphere. Their collective biomass outweighs all eukaryotes combined at least tenfold. More prokaryotes inhabit in a handful of fertile soil or the mouth or skin of human than the total number of people where lived. Prokaryotes thrive in habitats that are too cold, too hot, too salty, too acidic, or too alkaline for any eukaryote. Prokaryotes have even been discovered in rocks 2 miles below the surface of the earth. Prokaryotes are small and are unicellular. Some species may aggregate transiently or form true colonies, showing divisions of labor between specialized cell types. Most prokaryotic have diameters in the range of 1 to 5 nanometer compared to 10 to 100 nanometer for most eukaryotic cell. The largest prokaryotic discovered so far has a diameter of 750 nanometer. The most common shapes among prokaryotes are spheres, rods, and helix. Nearly all prokaryotics have a cell wall external to the plasma membrane. In nearly all prokaryotics, a cell wall maintains the shape of the cell, affords physical protection, and prevents the cell from bursting in a hypotonic environment. In a hypertonic environment, most prokaryotics lose water and plasmolyze like other walled cells. Severe water loss inhibits the reproduction of prokaryotics, which explains why salt can be used to preserve foods. Most bacterial cell wall contain peptidoglycan, a polymer of modified sugar cross-linked by short polypeptides. The walls of archaebacteria lack peptidoglycan. The gram stain is a valuable tool for identifying specific bacteria based on differences in their cell walls. Whereas gram-positive bacteria have large amount of peptidoglycan, it is absent in gram-negative bacteria. Many antibiotics, including penicillin, inhibits the synthesis of cross-linking in peptidoglycans, preventing the formation of functional wall, especially in gram-positive species. The cellular and genomic organization of prokaryotics is fundamentally different from that of eukaryotics. In prokaryotic genome of E. coli contains amount of 4 into 10 to the power of 6 base pairs. It is greater than 90% of the DNA encoded protein. It lacks a membrane-bound nucleus, that is, circular DNA and supercloid domain is present. Histones not present. Prokaryotic genome generally contains one large circular piece of DNA referred to as chromosomes. Some bacteria have linear chromosomes. Many bacteria have small circular DNA structures called plasmid, which can be swapped between neighbors and across bacterial species. Plasmid. The term plasmid was first introduced by American molecular biologist Joshan Ledbach in 1952. A plasmid is a separate form and can replicate independently of the chromosomal DNA. Plasmid size varies from 1 to over 1000 kilobase pairs. The cells of prokaryotes are simpler than those of eukaryotes in both internal structure and genome organization. Prokaryotic cells lack the complex compartmentalization found in eukaryotic cells. Instead, prokaryotics use specialized enfolded regions of the plasma membrane to perform many metabolic functions, including cellular respiration and photosynthesis. Prokaryotics have smaller, simpler genomes than eukaryotics. On average, a prokaryote has only about 1000 as much DNA as the eukaryote. In the majority of prokaryotes, the genome consists of a ring of DNA with a few associated proteins. The chromosome is located in the nuclear region. Prokaryotics may also have smaller rings of DNA called plasmid, which consists of only a few genes. They can survive in most environments without their plasmids because their chromosomes program all essential functions. Plasmid genes provides resistance to antibiotics, direct metabolism of usual nutrients, and other special contingency functions. Plasmids replicate independently of the chromosome and can be transferred between partners during conjugation. A great diversity of nutritional and metabolic adaptation have evolved in prokaryotics. Organisms can be categorized by their function based on how they obtain energy and carbon to build the organic molecules that make up their cells. Nutritional diversity is greater among prokaryotes than among all eukaryotes. Every type of nutrition observed in eukaryotics is found in prokaryotics, along with some nutritional modes unique to prokaryotes. Organisms that obtain energy from light are phototropes. Organisms that obtain energy from chemicals in environments are called chemotropes. Organisms that they need only carbon dioxide as a carbon source are autotropes. Organisms that require at least one organic nutrient such as glucose as a carbon source are called heterotropes. These categories of energy source and carbon source can be combined to group prokaryotes according to four major modes of nutrition: photoautotropes, chemoautotropes, photoheterotropes, and chemoheterotropes. Nitrogen is an essential component of protein and nucleic acid in all organisms. Eukaryotes are limited in the form of nitrogen they can use. In contrast, diverse prokaryotes can metabolize a wide variety of nitrogenous bases. Nitrogen-fixing prokaryotes convert nitrogen to ammonia, making atmospheric nitrogen available to themselves for incorporating into organic molecules. Nitrogen-fixing cyanobacteria are the most self-sufficient of all organisms. Prokaryotes were once thought of single-celled individuals. In some prokaryotic species, metabolic cooperation occurs in surface coating colonies known as biofilms. Cells in the colonies segregate signaling molecules to recruit nearby cells, causing the colony to grow. Once the colony is sufficiently large, the cells begin producing proteins that adhere the cells to the substrate and to one another. Channels in the biofilms allows nutrients to reach cells cells in the interior and allows waste to expelled. In some cases, different species of prokaryotes may cooperate. For example, sulfate-consuming bacteria and methane-consuming archaea coexist in ball-shaped aggregates in the mud of the ocean floor. The bacteria use the archaea waste products. In turn, the bacteria produce components that facilitate methane consumption by the archaea. Each year, the archaea consumes an estimated 300 billion kilograms of methane, a major greenhouse gases. Bacterial chromosomes in the nucleus. The nucleus is an irregularly shaped region within the cell of a prokaryote that contains all or most of the genetic material. In contrast to the nucleus of eukaryotic cell, it is not surrounded by a nuclear membrane. The genome of prokaryotic organism generally is a circular double-stranded piece of DNA of which multiple copies may exist at any time. The length of genome varies widely, but it generally at least a few million base pairs. The nucleus can be clearly visualized on a electron micrograph at high magnification, where it is clearly visible against the cytosol. Sometimes even strands of what is thought to be DNA are visible. The nucleus can also be seen under a light microscope by staining it with the fluorescent stain, which specifically stains DNA. The DNA intercalating stains DAPI and ethidium bromide are widely used for fluorescent microscopy of nucleoids. Experimental evidence suggests that the nucleus is largely composed of about 60% DNA plus a small amount of RNA and protein. The latter two constants are likely to mainly messenger RNA and the transcription factor proteins found regulating the bacterial genome. Proteins helping to maintain the supercoiled structure of the nucleic acid are known as nucleoid proteins or nucleoid associated proteins and are distinct from histones of eukaryotic nuclei. In contrast to histones, the DNA binding proteins of the nucleoid do not form nucleosomes, in which DNA is wrapped around a protein core. Instead, this protein often use other mechanisms such as DNA looping to promote companion. The genophore. A genophore is the DNA of a prokaryote. It is commonly referred to as a prokaryotic chromosome. The term chromosome is misleading because the genophore lacks chromatin. The genophore is compacted through a mechanism known as supercoiling, but a chromosome is additionally compacted through the use of chromatin. The genophore is circular in most prokaryotes and linear in very few. The circular nature of genophore allows replication to occur without telomeres. Genophores are generally of much smaller size than eukaryotic chromosome. A genophore can be as small as 5,80,073 base pairs as in Mycoplasma genitalium. Many eukaryotes carry genophore in organelles such as mitochondria and chloroplast. These organelles are very similar to true prokaryotes. Eukaryotic genome organization. Eukaryotic genome is linear and conforms the Watson and Crick double helical structural model. Embedded in nucleosome, complex DNA and protein structure that pack together to form chromosome. Eukaryotic genome have unique features of exon, intron organization of protein coding genes representing coding sequences and intervening sequences that represent the functionality of RNA part inside the genome. The genome of each cell contains 1.35 into 10 to the power of 7 base pairs. A small fraction of the total DNA encodes protein. Many repeats of non-coding sequences are found in the DNA. All chromosomes are contained in the membrane-bound nucleus. DNA is divided into 2 or more chromosomes. A set of 5 histones are present. DNA packing is and gene expression is regulating. The study of chromosome, the structure and their inheritance is known as cytogenetics. Each species has a characteristic number of chromosomes and this is known as karyotype. In bacteria, the number of chromosomes are 1, fruit fly 8, garden pea 14, yeast 16, frog 26, cat 38, fox 34, mouse 40, rat 42, rabbit 44, human 46 and then chicken 78 number of chromosomes are present. Prior to 1950s, it was believed that humans had 48 chromosomes, but in 1956 it was confirmed that each human cell has 46 chromosomes. On the chromosomes, the genes are situated in a linear order. Each gene has a precise position or locus. The size of bacterial chromosomes ranges from 0.6 to 10 megabase pairs, and the size of archaeal ranges from 0.5 to 5.8 megabase pairs, whereas eukaryotic chromosomes ranges from 2.9 to 4000 megabase pairs. Eukaryotic genome is more complex, which contains nuclear, mitochondrial, plasmids. The human genome contains a genetic material in the human is stored in two organelles, nucleus about 3200 megabase pair and mitochondria 16.6 kilobase pair. Human chromosomes are not of equal size. The smallest chromosome is 21 and the largest chromosome 1. Only a very small amount of human DNA is responsible for the differences among human, indeed among all the organisms. The number of genes present in the human genome is at least 1 lakh base pairs. However, the number of protein encoded gene is only 20,000 to 25,000. Mitochondrial DNA. Mitochondrial DNA is a single double-stranded circular molecule. There are several copies in each mitochondria, and there are many mitochondria in each of your cells. Mitochondrial DNA is similar to prokaryotic DNA. There are no histones or any other protein associated with mtDNA. The gene contains no introns, maternal inheritance because it is a highly oxidizing environment. It has a much higher rate of mutation than nuclear DNA. The genes in the mitochondrial DNA code for mitochondrial ribosomes and transverse RNAs. Some genes code for polypeptide subunits of the electron transport chain common to all mitochondria, and some of the genetic information on the chromosomes. One gene hypothesis summarizes that a gene is a stretch of DNA coding for one or more isoforms of a single enzyme. One gene, one polypeptide hypothesis. A gene is responsible for the production of a single polypeptide. Many genes, one protein. Example, hemoglobin requires different globin genes. Configuration of eukaryotic genome. The configuration of eukaryotic genome includes protein coding region, gene regulating region, gene related sequence, and intergenic DNA or extragenic DNA, which includes low copy number and moderate or high copy number repetitive sequence. First of all, gene and gene related sequences in eukaryotes. Protein coding regions, that is exons. Protein coding sequences are the DNA sequences that are transcribed into mRNA, later translated into proteins. The complete protein coding sequence capacity of genome is contained within the exomes. These are the reading frame that has the potential to code for the protein or peptide. It is stretch of codons that do not contain a stop codon. An AUG with the ORF may indicate where translation starts. Gene regulating sequences in eukaryotes. Promoters are combination of short sequence elements usually located in the immediate upstream region of a gene, often within 200 base pair of the transcription start site, which serve to initiate transcription. They can be subdivided into different components. The core promoter directs the basal transcription complex to initiate transcription of the gene. In the absence of additional regulatory elements, it permits constitutive expression of the gene, but at very low levels. Core promoter elements are typically located very closely to the transcription initiation site at about nucleotide position -40 to +40. The proximal promoter region is the sequence located immediately upstream of the core promoter, usually from -50 to -200 base pairs. Promoters elements found further upstream would be said to map to distal promoter region. They include GC boxes. Enhancer are positively transcriptional control elements which are particularly prevalent in the cells of complex eukaryotes such as mammals, but which are absent or very poorly represented in some eukaryotes such as yeast. They serve to increase the basal level of transcription, which is initiated through the core promoter elements. Their function, unlike those of the core promoter, are independent of both their orientation and to some extent the distance from the genes they regulate. Enhancers often contain within a span of only 200 to 300 base pairs. Silencers. Silencers serve to reduce transcriptional levels. Two classes have been distinguished: classical silencers, negative regulator elements. Classical silencers are position independent elements that directly and active transcriptional repression mechanisms, whereas negative regulatory elements are the position dependent elements that result in passive repression mechanism. Silencer elements have been reported in various positions close to the promoter, some distance upstream, and also within the introns. Boundary elements. Boundary elements are insulators or region of DNA often spanning from 0.5 kilobase pair to 3 kilobase pair, which function to block the spreading of the influence of agents that have a positive effect of transcription, that is enhancers, or negative one, that is silencers, heterochromatic like repressive effects. Response elements. Response elements modulate transcription in response to specific external stimuli. They are usually located a short distance upstream of the promoter elements. A variety of such elements respond to specific hormones or to intracellular second messengers such as cyclic adenosine monophosphate. Non-coding functional RNA. A non-coding RNA is an RNA molecule that is not translated into protein. Less frequently uses synonyms are non-protein coding RNA, non-messenger RNA, and functional RNA. The DNA sequence from which a functional non-coding RNA is transcribed is often called as RNA gene. Introns. The term introns refers to both the DNA sequence within a gene and the corresponding sequence in RNA transcripts. At least 4 distinct classes of introns have been identified. Introns in nuclear protein coding genes that are removed by spliceomes. Introns in nuclear and archaeal transfer RNA genes that are removed by proteins. tRNA introns. Self-splicing group 2 introns that are removed by RNA catalase. Group 3 introns are proposed to be a fifth family, but little is known about the biochemical apparatus that mediates that splicing. They appear to be related to group 2 introns and possibly to spliceomal intron. Gene fragments. Gene fragments are pieces of genes containing only the exons. They are composed of complementary DNA. Pseudogenes. Pseudogenes are dysfunctional relative of genes that have lost their gene expression in the cell or their ability to code protein. Pseudogenes often result from the accumulation of multiple mutations within a gene whose product is not required for the survival of organism. Depending on their DNA sequence, pseudogenes are mainly of two types: processed pseudogene and unprocessed pseudogene. The processed pseudogene, they have all the normal parts of protein coding gene but are originally thought to be long non-coding RNA. Long non-coding RNA are a type of non-coding RNAs that exceed 200 nucleotides in length. They are relatively abundant component of the mammalian transcriptome and have implicated in several cellular functions, including the regulation of gene transcription through the requirement of chromatin modifying enzymes. Intergenic DNA. An intergenic region is a stretch of DNA sequence located between genes. Intergenic regions are subset of non-coding DNA. Occasionally, some intergenic DNA acts to control genes nearby, but most of it has no currently known function. It is sometimes referred to as junk DNA. In humans, intergenic regions comprise about 75% of the genome, whereas this number is much less in bacteria, that is 50%, and yeast 30%. Intergenic regions are different from intragenic regions, which are short non-coding regions that are found within the genes, especially within the genes of eukaryotic organism. DNA transposons. The cut and paste transposition mechanism of class 2 transition elements does not involve an RNA intermediate. The transposition are catalyzed by several transposase enzymes. Some transposase non-specifically bind to any target site in DNA, whereas others bind to specific DNA sequence targets. DNA transpones have terminal inverted, repeated, and encode a transposase that regulate transposition. They account for close to 3% of the human genome. Virtually all all the resident human DNA transposon sequences are no longer active. They are therefore transposon fossils. DNA transposons tend to have short lifespans within a species, unlike some of the other transposable elements such as long interspersed nuclear elements. These are the organization of genome in prokaryotic and eukaryotic organisms. Thus, we will conclude this episode. We will see the new topic on the next episode, that is enzymes involved in DNA replication. Thank you.